Right, let's get into it. Today we're tackling a massive topic in immunology antibodies. You've probably heard the term thrown around a lot, especially in the context of infections and vaccines. But what are they really? Think of them as your body's highly specialized security team. They are proteins produced by a type of white blood cell called a B lymphocyte or B cell. Their primary job is to identify and neutralize foreign invaders like bacteria, viruses, and other pathogens. They are a critical part of your adaptive immune system, which is the part of your immunity that learns and remembers specific threats, allowing for a faster and stronger response the next time you encounter the same bug. Now here's a key bit of terminology you need to get straight from the start. Antibodies are also known as immunoglobulins. You'll see this written as Ig. So, when you hear someone say immunoglobulin G or see IgG written in a patient's chart, they are simply talking about a specific type of antibody. Using the terms interchangeably is common practice in medicine, so it's essential to recognize both. These proteins are globulins, a category of proteins found in your blood plasma, and because they are involved in the immune response, they get the prefix immuno. It's a logical name, but antibody is definitely the more common term you'll hear in everyday conversation. Imagine a microscopic Y-shaped grappling hook floating through your bloodstream. That's a pretty good mental picture of a basic antibody molecule. This unique shape is not just for show, it's fundamental to how they function. Each antibody is designed with incredible specificity. It's like having a key that only fits one specific lock. The lock in this case is a unique molecule on the surface of a pathogen called an antigen. When an antibody finds its matching antigen, it binds to it tightly. This binding action is the first step in a cascade of events designed to eliminate the threat from your body, a process we'll dive into much deeper later on. The sheer diversity of antibodies your body can produce is staggering. We're talking about millions, potentially billions of different variations. This incredible range is necessary because there are countless different pathogens in the world, each with its own unique set of antigens. Your immune system has to be prepared for almost anything it might encounter. This is why when you get sick with a new virus, it takes time for your body to mount an effective defense. Your B cells need time to develop and produce the exact right antibody to fight that specific invader. Once they do, you've got a powerful, tailor-made weapon at your disposal. Alright, so we know antibodies or immunoglobulins are key players, but it's not a one-size-fits-all situation. Your body produces 5 distinct classes or isotypes of immunoglobulins. Think of them as different branches of your special forces, each with its own unique skills, deployment areas, and missions. These 5 classes are immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, and immunoglobulin D. A simple way to remember them is with the acronym GAMED. Each class has a different structure and is found in different parts of the body, allowing them to tackle threats in a coordinated and highly effective manner. Let's do a quick roll call. First up is IgG, the most abundant antibody in your blood and tissues, making up about 75 to 80% of the total immunoglobulins in your serum. Next is IgA, the main protector of your mucosal surfaces. Think of the linings of your gut, respiratory tract, and even in your tears and saliva. Then we have IgM, the big guy, which is the first antibody to appear in response to an initial exposure to an antigen. After that is IgE, the one famous for its role in allergic reactions and fighting off parasitic worms. And finally, there's IgD, the most mysterious of the bunch, which is found in small amounts and acts primarily as an antigen receptor on B cells. Understanding the roles of these 5 classes is crucial for clinical practice. When a lab report comes back showing elevated levels of a specific immunoglobulin, it gives doctors important clues about what's happening inside the patient's body. For example, high levels of IgM often suggest a recent or current infection, as it's the first responder. In contrast, high levels of IgG might indicate a past infection or a successful vaccination, representing the long-term memory response. Similarly, an unusually high IgE level would immediately make a clinician think about allergies or a possible parasitic infection. They are diagnostic markers that tell a story about the patient's immune status. The structural differences between these classes are what allow them to perform such varied functions. For instance, IgM circulates as a large pentamer, meaning 5 Y-shaped units are joined together, making it incredibly effective at grabbing onto pathogens early in an infection. IgA, on the other hand, often exists as a dimer, which is perfect for its job of patrolling the vast mucosal surfaces. IgG, IgE, and IgD typically exist as simple monomers, single Y-shaped units. We are going to break down these structures and functions in much more detail so you can see exactly how each member of the gamed family contributes to keeping you healthy. So we've established that the basic antibody is a Y-shaped protein. Now let's zoom in and look at its architecture because understanding its structure is fundamental to understanding its function. Each antibody monomer is built from 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. Imagine the Y shape. The 2 heavy chains are the larger proteins that form the entire stem and the inner part of the arms of the Y. The 2 light chains are smaller and are attached to the outer part of the arms of the Y. These chains are held together by strong chemical bonds called disulfide bridges, creating a stable yet flexible unit. This is where it gets really interesting. Each of the heavy and light chains is divided into 2 distinct regions, a constant region and a variable region. The constant region, as the name suggests, is pretty much the same for all antibodies within the same class. For example, all IgG antibodies will have a very similar constant region, which is different from the constant region of all IgA antibodies. This part of the antibody, located in the stem of the Y and the lower part of the arms, is what determines the antibody's class and its general effector function, that is, how it signals to other parts of the immune system. Let's call the constant region the handle of the antibody. It's the part that other immune cells, like macrophages or mast cells, can grab onto. This region, also known as the Fc region, dictates the mechanism that will be used to destroy the antigen. For instance, the constant region of an IgG antibody might be designed to bind to receptors on a phagocyte, a cell that engulfs and digests pathogens. In contrast, the constant region of an IgE antibody is designed to bind to receptors on mast cells, triggering the release of histamine during an allergic reaction. So the constant region defines how the antibody will fight. To put it simply, you can think of an antibody molecule as a highly specialized tool. The variable region, which we'll discuss next, is the working end of the tool, the part that is uniquely shaped to interact with a specific target. The constant region is the handle of that tool. While the working end changes for every single job, every specific antigen, the handle is one of 5 standard types: IgG, IgA, IgM, IgE, or IgD. This standard handle allows other workers, other immune cells, to easily recognize the tool and know how to assist in the job of eliminating the pathogen. This dual component structure is a masterpiece of biological engineering. Now let's focus on the most dynamic part of the antibody, the variable region. This region is located at the tips of the 2 arms of the Y. Unlike the constant region, the amino acid sequence in the variable region is, well, highly variable. This is the part of the antibody that is exquisitely specific for its target antigen. Each B cell, through a remarkable process of genetic rearrangement, crafts a unique variable region for the antibodies it will produce. This means that an antibody designed to fight the chickenpox virus will have a completely different variable region from an antibody designed to fight the tetanus toxin. This specificity is the cornerstone of adaptive immunity. The variable regions of 1 heavy chain and 1 light chain combine to form a single antigen binding site. Since an antibody monomer has 2 arms, it has 2 identical antigen binding sites. This part of the antibody is also known as the Fab region, which stands for fragment antigen binding. The shape and chemical properties of this site are precisely complementary to a specific part of an antigen, known as an epitope. The fit is so precise, it's often compared to a lock and key. This binding is not just a loose attachment, it's a strong, non-covalent bond that holds the antibody firmly to its target. The incredible diversity of the variable region is what allows your immune system to recognize a virtually limitless number of different antigens. The genetic shuffling that creates these regions means your body can generate an antibody for a pathogen it has never even encountered before. When a B cell with a perfectly matching receptor, a surface-bound antibody, finally bumps into its specific antigen, it triggers that B cell to activate, multiply, and start churning out huge quantities of that specific antibody. This clonal selection ensures that you produce a massive targeted army against the invader, rather than a scattered, ineffective response. Having 2 antigen binding sites on a single monomer, a property known as bivalency, significantly increases the overall binding strength or avidity of the antibody to its target. Think about trying to grab something with 1 finger versus using 2 fingers. the two-point contact is much more secure. This is particularly important when dealing with pathogens that have multiple identical epitopes on their surface. An antibody can potentially bind to two separate epitopes on the same pathogen, or cross-link two different pathogens together, which is a key mechanism for clumping them up and marking them for destruction, a process we'll explore shortly. Let's start with the first responder, IgM. When your body encounters a pathogen for the very first time, the first antibody class to be produced in the primary immune response is IgM. Think of IgM as the SWAT team leader, the first one on the scene to assess the situation and start taking action. Structurally, IgM is a giant. It exists as a pentamer, which means 5 individual Y-shaped antibody units are linked together in a star-like shape by a central J chain. This large multi-unit structure gives it a total of 10 antigen-binding sites. This makes IgM incredibly effective at agglutination, which is a fancy word for clumping pathogens together. IgM is a master of agglutination and complement. With its 10 binding sites, a single IgM molecule can grab onto multiple pathogens at once, like a microscopic ball and chain. This clumping action makes it much harder for bacteria or viruses to move around, infect cells, and cause damage. More importantly, it creates a large, easily recognizable target for other immune cells like macrophages to find and engulf. Furthermore, IgM is extremely efficient at activating the complement system, a powerful cascade of proteins that helps to destroy pathogens. Its large size, however, means it's generally confined to the bloodstream and doesn't easily cross into tissues, limiting its field of operation. Then comes the main workhorse, IgG. Following the initial IgM response, the B cells undergo a process called class switching and start producing IgG. IgG is the most abundant antibody in your blood and is the star of the secondary immune response. That's the response you get upon a second encounter with the same pathogen. It exists as a simple monomer, a single Y-shaped unit. While it only has 2 binding sites compared to IgM's 10, the binding affinity of these sites is often much higher, meaning it binds more tightly to its target antigen. IgG is smaller and more versatile than IgM, allowing it to easily leave the bloodstream and travel into the tissues to fight infections wherever they might be. IgG provides long-term memory and protects the fetus. IgG is the antibody of immunological memory. Its levels remain elevated long after an infection has been cleared, providing you with long-term protection. This is the antibody that vaccines aim to generate. Crucially, IgG is the only antibody class that can cross the placenta from mother to fetus. This provides the baby with passive immunity, protecting it from various infections for the first few months of life until its own immune system matures. IgG is a jack of all trades. It can neutralize toxins, immobilize bacteria, tag pathogens for destruction by phagocytes, a process called opsonization, and also activate the complement system, though not quite as efficiently as IgM. Now let's talk about IgA, the second most common antibody in the body. While IgG dominates the bloodstream, IgA is the undisputed champion of the mucosal surfaces. Your body has vast areas of mucosa lining the respiratory tract, the gastrointestinal tract, and the urogenital tract. These are the primary entry points for pathogens, and IgA is stationed there like a bouncer at a club. It's found in secretions like saliva, tears, sweat, and mucus, as well as in breast milk. Its main job is to prevent pathogens from ever getting a foothold and attaching to the surfaces of your cells in the first place. In secretions, IgA typically exists as a dimer, meaning 2 Y-shaped monomers are joined together by a J chain, similar to the one in IgM. This dimer is also wrapped in a protein called the secretory component. This special component acts like a protective shield, preventing the IgA molecule from being broken down by the harsh digestive enzymes found in the gut. This robust structure allows it to survive and function effectively in these challenging environments. By binding to bacteria and viruses on mucosal surfaces, IgA neutralizes them before they can even enter the body's tissues, acting as a crucial first line of defense. It's also the main antibody passed to infants through breastfeeding, protecting their gut. Next up is IgE. This antibody is found in very low concentrations in the blood of most people. It's a monomer, just like IgG. However, its name is most famously, or perhaps infamously, associated with allergic reactions. The constant, or FC region of IgE is designed to bind with extremely high affinity to receptors on the surface of mast cells and basophils. These are immune cells packed with granules containing powerful inflammatory chemicals, including histamine. When an allergen, like pollen or peanut protein, enters the body of a sensitized person, it cross-links the IgE molecules sitting on these mast cells. This cross-linking of IgE acts like a trigger, causing the mast cells to degranulate, that is, to release their contents in a rapid, explosive burst. The release of histamine and other mediators causes the classic symptoms of an allergic reaction: vasodilation, which leads to redness and swelling, increased vascular permeability, causing runny noses and watery eyes, and smooth muscle contraction, which leads to bronchoconstriction and asthma. While it causes misery for people with allergies, the original evolutionary purpose of IgE is thought to be defense against parasitic worms. The inflammatory reaction it causes can help to physically expel these large parasites from the body. Finally, we come to the last member of our gamed lineup, immunoglobulin D, or IgD. Of all the antibody classes, IgD is the one we understand the least about. It's found in extremely low levels circulating in the blood, where it exists as a monomer structurally similar to IgG. For a long time, its function was a complete puzzle to immunologists. We knew it existed, but what it actually did in the grand scheme of the immune response was unclear. It doesn't seem to be very good at activating complement, and it doesn't bind to the standard receptors on phagocytic cells, so its role as a direct effector molecule is limited. The primary location where we find IgD is not floating free in the plasma, but on the surface of naive B lymphocytes, that's B cells that have not yet been activated by an antigen. Here, alongside IgM, it functions as a B cell receptor, or BCR. Think of the B cell receptor as the cell's personal scanner. The B cell patrols the body, and its surface-bound IgM and IgD molecules are constantly feeling for an antigen that matches their specific variable region. When the right antigen comes along and binds to these receptors, it sends a powerful signal into the B cell, telling it that it's time to get to work. The binding of an antigen to the IgD on a B cell's surface is a critical step in initiating the adaptive immune response. This signal, often with help from another immune cell called a T helper cell, triggers the B cell to undergo clonal expansion and differentiation. This means it starts to divide rapidly, creating an army of clones, and matures into either a plasma cell, which is a factory for secreting antibodies, or a memory B cell, which will provide long-term immunity. So, while circulating IgD doesn't seem to do much fighting itself, surface-bound IgD plays a crucial role as a gatekeeper for B cell activation. Recent research suggests that IgD might do more than just act as a simple on switch. It appears to work in concert with IgM to fine-tune the B cell response. While IgM is great at signaling for a strong initial activation, IgD might be involved in sending signals that promote B cell survival and differentiation into plasma cells. Some evidence also suggests it may play a role in activating basophils and mast cells to produce antimicrobial factors, contributing to respiratory immune defense. IgD may be the quiet one in the family, but it's an important team player that helps ensure the entire immune operation runs smoothly from the very beginning. We've spent a lot of time on the who and what of antibodies. Now let's get to the how. How does an antibody actually get rid of a pathogen once it has latched on? The mechanisms of antibody action can be broadly divided into two main categories. The first is direct action, where the binding of the antibody itself has a direct effect on the pathogen. The second, and arguably more powerful category, involves the antibody acting as a flag, or a tag, recruiting other parts of the immune system to do the dirty work. This indirect action often involves the activation of the complement system, which we'll cover in the next section. Let's start with the direct mechanisms. The simplest is neutralization. Many pathogens, particularly viruses and bacterial toxins, need to bind to specific receptors on your own cells to cause harm. An antibody can physically block this from happening. By binding to the critical sites on a virus or toxin, the antibody effectively neutralizes it, rendering it harmless because it can no longer interact with your cells. Another direct mechanism is agglutination, which we mentioned with IgM. By using their multiple binding sites to clump pathogens together, antibodies can immobilize them and prevent them from spreading through tissues, making them an easy target. A similar process to agglutination is precipitation. This applies to soluble antigens like toxins or other proteins dissolved in your body fluids, rather than whole cells like bacteria. When antibodies bind to these soluble molecules, they can form large, insoluble immune complexes that precipitate out of the solution. Just like with agglutination, this makes the antigens much easier for phagocytic cells to find and clear away. These direct actions, neutralization, agglutination, and precipitation, are effective ways to contain a threat, but they often rely on the second category of mechanisms to finish the job. This brings us to the indirect actions, where the antibody acts as an intermediary. One of the most important of these is opsonization. The word comes from a Greek term meaning to prepare for eating, and that's exactly what it is. Phagocytic cells like macrophages and neutrophils are the garbage collectors of your immune system, responsible for gobbling up pathogens. While they can recognize some pathogens on their own, the process is much more efficient when the pathogen is coated with antibodies. The phagocytes have Fc receptors on their surface that bind to the constant or Fc region, the handle of the antibodies, specifically IgG. This coating or opsonization makes the pathogen irresistible to the phagocyte, marking it clearly for destruction. Sometimes just tagging a pathogen isn't enough. For the toughest jobs, antibodies call for heavy artillery, and in the immune system that means activating the complement system. Complement is a collection of over 30 proteins circulating in your blood, usually in an inactive state. When activated, these proteins work together in a precise enzyme-driven cascade to obliterate pathogens. The classical pathway of complement activation is triggered directly by antibodies, specifically IgM and IgG, that are bound to the surface of a pathogen. This is one of the most powerful ways antibodies eliminate threats. It all begins when the first complement protein, C1, recognizes and binds to the Fc regions of two or more antibodies that are attached to a pathogen. IgM, with its 5 Fc regions clustered together in a pentamer, is exceptionally good at this. IgG can also do it, but it requires at least 2 IgG molecules to be close enough together on the pathogen's surface. This binding of C1 is the spark that ignites the entire complement cascade. C1 becomes an active enzyme which then cleaves and activates the next complement protein, which in turn activates the next and so on, in a rapidly amplifying chain reaction. This cascade has several powerful outcomes, but the most dramatic is the formation of the membrane attack complex, or MAC. As the cascade progresses, a group of complement proteins, C5b, C6, C7, C8, and C9, assemble themselves on the pathogen's surface. They come together to form a pore, or a channel, right through the pathogen's cell membrane. Think of it as punching a hole in the side of a ship. With this hole in its membrane, the bacterium or infected cell can no longer maintain its internal stability. Water and salts rush in, causing the cell to swell and burst, a process called cytolysis. It's a direct and brutally effective way to kill an invader. So as you can see, antibodies are much more than just simple binding proteins. They are the conductors of a complex orchestra of immune defense, from directly neutralizing threats to tagging them for phagocytes and unleashing the destructive power of the complement system. They are central to protecting you from disease. Understanding their structure, the different classes, and their mechanisms of action is fundamental to your journey in medicine. I hope this deep dive has been helpful. If you want more clear, no-nonsense content like this to help you ace your exams and become a better clinician, make sure you subscribe to Joen Academy. We're breaking down complex medical topics every single week. Stay curious, and I'll see you in the next video.